actually Applegate. We're just kind of trying to get an idea of who's on the website so that we know who's in session B, because we have myself and Richard Wakeford being the co-chairs of Group B, and we have two rapporteurs who are mentees for Task Group 121. We have Amelie and Ludmilla, thank you. I'm having a brain cramp. So on the web, we have Iggy, Klaus, Ahmed, and Sarah. Sarah is also a mentee for the task group. So thank you all for being here. Can you hear me all right?
No.
No?
Please go closer to the mic. Maybe that helps.
Can you increase the volume?
Yes.
Okay, we're going to increase the volume on the microphone.
Yeah, that's fine. That's better.
Great, thanks, Klaus. Okay, now I'm going to just do a short presentation that, and we're going to get started on the presentation so that we can have time to have some discussion today. We're going to we have more presentations. That's some of this may have been covered, but now you can minimize that by the other presenters. But I did not know exactly what they were going to present. Are you guys okay?
I don't know names. If you could repeat your names.
No, yeah, Iggy. So there.
Just quickly, Sila Rudas?
Sarah Dumit.
Sila Rudas is here, okay.
Oh, okay. Sorry. I should go around the room then if you want first. I'm sorry.
Yeah, okay, great.
Okay.
Stephanie Grissom? Online?
Yes.
Stefan, yeah, sorry.
Augusto, before he was, but now he's
Oh, that's a good point. He's not here. He must have chosen
here, no?
Eric
Rizzo?
and
She might be online.
Rizzo?
no, I don't see her on the website.
In Japan, so.
Yeah, it's true.
Ignacia? Yes, she was here. Sarah Batut? No, I haven't seen her, no? Marta Painsko? Marta, online? Sarah Dumit, she was before.
Sarah is on.
Omid? Also. Okay. Thank you very much.
Thank you. And I think, let's see. Right, okay. And we'll try to track, each time. Yeah, thanks. Okay, so quickly, I wanted to say something about epigenetics, which you see on this is a slide of my twins. They're identical, and when they were young. They're now adults. But there's no doubt that epigenetics plays a strong role, I think. And I'm not convinced that it doesn't play an ongoing role, although this is anecdote and not evidence. Because the one on the left, I was told that very early in gestation on ultrasound that I had twin transfusion, and I'm a twin. So it's interesting, so that it's interesting just by happenstance that I had these identical twins that the one on the left had more blood flow and so more fluid in the amniotic sac than the one on the right. So he was born bigger, and he ended up growing faster, and he just had a different personality than the other one, but they are very close even to this day. And on tests in school, they track identically. I mean, they were within a few points. They may have a slightly different personality, but they both majored in computer science and work in computer science in college, after college, and track very close in math and language. So anyway, that's interesting. I gotta go to the next. How do I go forward? Do I have to?
Just press the right one.
I thought I did.
Just.
Oh, maybe it's like there.
I will change the slide and then.
Okay. Thank you, thank you. It's just, I only have a few slides, but I wanted to cover a few. I'm not a biologist and I'm not a geneticist, so here I am. And I'm covering some of the, more of the applications viewpoint, and some of this is already covered. You know, this preconception concept is going to be covered by different groups and different speakers, but we know that there's no human germline, at least we don't think there's human germline mutation, but I always wonder if we're unable to detect it, so prudence dictates that we be careful. And I want to point out that when we think about temporary and permanent sterility, it depends on both the sex and the age. So for example, younger women require lower radiation dose to have sterility. So we have to be careful about what we're talking about. And then tissue effects also depends very much on timing of the radiation to the fetus. And then cancer, I think, Richard, you mentioned this. What we tell people is that it doesn't matter what the fetal age is, the effect is the risk is the same. But in fact, if you look at our ICRP publications, we've said that there may be, or if in animal studies, there may be a higher risk late in gestation. And so I'm raising it. I know, Richard, you raised it on your slide. Next slide. Now we have to know baseline effects because we have to find a signal underneath all of these baseline effects. 15% of pregnancies result in spontaneous loss, usually before the woman knows she's pregnant. There are 3% of births have major malformations, 4% have minor, and some of these we never even know about until autopsies reveal something. And 0.4% of children have intellectual disability of some form, 4% of microcephaly. So you can have microcephaly and be normal, or you can have...
The sound is disappearing gradually.
I'm sorry, I think I turned my
thank you.
head. I have to remember to keep the microphone with my head. I'm just trying to describe that you can have microcephaly and have a
Thank you.
normal intellectual ability, or you can have an intellectual disability and have microcephaly. So one, they're not always connected. And next slide. This one was shown by Prakash, I think, so we don't have to go through this, but it is the most important slide in the whole talk. Understanding the timing of the radiation exposure to the embryo or fetus is everything. It's everything when we're trying to counsel the family about what to do or what not to do, you know, to do nothing. And most of the time, like in medical imaging, we tell them, do nothing. You know, the risk is tiny. That doesn't mean they're going to take our advice, as we know from the Chernobyl, but it's what we try to tell people. And we do need to understand the all or none phenomenon, that in the first 14 days, there's fetal loss at 150 milligray or higher, but otherwise there are different effects at different doses. Next slide. So the most critical malformation period is and the major organogenesis period, which is 2 to 7 weeks post-conception, but it takes a relatively higher dose than the most critical radiosensitive period, which is 8 to 15 weeks post-conception. And that threshold is 100 milligray for neuropathology, growth retardation, and a few other things. But these are just numbers for thresholds that we've determined where we act very prudently setting these thresholds. And then later in pregnancy, when the organs have been formed but they're growing, it takes a relatively high dose to see change. Next slide.
Is it more or equal or less equal?
Greater.
Less.
Greater to see change. You don't see change.
So no change if great.
unless you, yeah, once the organs are formed and the fetus is just growing, you don't see the congenital malformations that you see in that critical organogenesis period. Now, you can see smaller babies, right? There's a risk of growth retardation overall, and there's certainly a cancer risk. It's small. What?
I'm speaking about doses.
Yeah. Oh, yeah. The higher the dose, the higher the risk. There's always.
But if you go the slide,
Okay.
the whole slide, it was higher, no, this known or
Can you go back one? Can you go back one?
because I was just bouncing away.
Sorry.
So just pre-implantation. All non pregnancy loss, it was higher or less?
I'm sorry. I'm not following you.
Just slide. Pre-implantation. Just
Pre-implantation, all or none. So if doses are, the threshold dose is 150 milligrams, felt to be 150 milligrams for human embryonic loss in that pre-implantation period. Okay?
But before, I mean, formulation was a
All right, next slide. Thanks.
Next or this one?
This one. One of the things we also know about from medical literature that isn't common in the RP literature is therapeutic abortion used radiation in the 1930s. And this is from Bushberg's medical physics textbook. It took 3 to 5 gray over 2 days, so it was a split dose. And these are in women who were felt not to be suitable to have surgery. So this was performed. It's certainly not done today, to the best of my knowledge. And so this is kind of a dose range for the therapeutic abortion, and in the not in those patients. There's also some literature that I was able to find when I did a review of doses in radiotherapy patients in the 1950s and 60s. And these were usually cervical cancer patients and lymphoma. And in a case series that Bowerman reported, and there were some other references that I couldn't get from his paper, these fetuses were in second and third trimester, and they got up to 8.5 gray. Yeah, I mean, this is, pretty amazing doses, and they had reports of being normal at birth. And some of them were normal in early childhood. So they were able to follow them at least until birth, and sometimes even beyond, for a few years without cancer or congenital malformations. So, there's a range of tolerance. And what we define as lethality. So yes, there may be a threshold, but there's quite a range. Next slide. And then the last thing I wanted to say is that we have an increase, as I think Prakash said, that there's quite an increase in use of imaging that uses ionizing radiation and non-ionizing radiation in pregnant women. So there's a paper from about 10 or 15 years ago that showed the average CT doses and the range, and it's from Rhode Island. And then there's a more recent 20-year period of 1996 to 2016 in the US and Canada. It was in JAMA Network Open, published in 2019, and it showed a pretty surprising 3.7-fold increase in CT imaging in the United States and doubling in Canada in that period. And what I think was really interesting to me was that almost 1% of pregnant women in the United States got CT scans. And, mostly that's attributable to trauma, but also headache and other conditions that women have that are pregnant. They have more, lots of conditions that are of concern, like pulmonary embolism and AV malformations and tumors. 1 in 1,000 get tumors. Next slide. I mentioned this. I'll mention one thing that besides Down syndrome, there's one report of BRCA1, BRCA2 children who, when they looked back and found chest x-rays from childhood, they linked that to breast cancer in adult women. Next slide. So I put forward my research questions, and I would say, are we missing subtle IQ effects from relatively high radiation? We don't know. Second, what are the cancer risks, as I mentioned before? We haven't linked any real cases, although we've seen a lot of epidemiological risk raised. And then ICRP publication 79 talked about genetic susceptibility to cancer, and they raised this question of tumor suppressor gene deficiency and reduction of tumor latency. So should we be looking for more of that? And then I'll leave you with this new ways of looking at organelles, by growing organelles, using human organelles like this blastoid on the image you see from a Nature publication. We can at least maybe look at embryos and look at radiation effects. This paper looked at implantation and embryonic development up to 14 days, which is the ethical limit of research. And the same, I can pass around this paper, the same is true for an ovioid that is now available for research use. It's they can take a stem cell, a pluripotent stem cell, a human stem cell, and do the same. Next slide, I think that's it. Yeah, so better. Thank you, and we should go on to the next speaker.
So I'm Ludmila Lutsko, and before when I worked on it, I was working in it's global together with Cardis, and this actually study was performed together with Sergei Igumnov and Vladimir Drozdovich, and it's based on a cohort from Belarusian in utero exposed. And the title of the presentation, relationship between thyroid hormone levels, radiation dose, and IQ in children exposed in utero by the Chernobyl accident and controls. So actually, this study, it was like exploratory, preliminary study performed just before pandemic. And here you can see, maybe all of you know about Chernobyl, but I just briefly, to put you on a set, it was, the Chernobyl accident happened 26th of April 1986 in former USSR, and actually in the territory of Ukraine current now, but it's more on the border with Belarus. And on the left, you see the picture of, you see here the picture of like representing these disasters. You see firemen and everybody, and here a woman who is breasting her child. And the main exposure from this accident was radioactive iodine-131 and cesium-134 and 137. And actually, also, though iodine was radioactive element which has a very short half-life time, so it's about around 8 days, but it was the most impactful after this accident, if you know, especially for Belarus, because this country, they have deficiency in iodine, stable iodine. So when you are have your thyroid has a deficiency of stable iodine, of course you are taking the radioactive one. And for children especially, it was for more vulnerable because with products with consumption of milk and dairy and some leaf vegetables, they were consuming more. Here, just to show the map of distribution of contamination, you see here for map for cesium 137. So Chernobyl is here somewhere. No, you see in the red, and it's actually the border with from Ukraine and Belarus. And you can see that the wind was blowing on the north, so Belarus was affected, Gomel, Mogilov, and here Bryansk region of Russia. And this map for iodine 131 after the Chernobyl accident, and the same you have here like two plumes, and here also Gomel and Mogilov are the most affected by concentration. So previous studies already done on this cohort showed that thyroid function can be altered by both exposure radiation, and in other studies I revised by Israeli scientists, they also showed how stress can cause on the biological level, oxidative level, with releasing free radicals similar to radiation. That's why I was pointing that it's sometimes difficult to separate both effects. And in this study, the aim of the study was to explore the relationship between thyroid hormones levels, dose, and general IQ level in two groups, in utero exposed and the control group. So in totally we had 500 persons, 250 persons were in utero exposed, born between May 1986 and February 1987, randomly selected from the state registry cohort children exposed to radiation following the Chernobyl accident. And here you can see that their mothers were lived in highly contaminated settlements with the solid deposition densities shown here on the slide. And the control group were also randomly selected 250 persons, like matched in sex and age. They were born the same dates, but their mothers were living or residing in relatively clean territories of Belarus, with you see much lower contamination of cesium. And for dosimetry, here Vladimir Drozdovich was doing, thyroid doses from iodine intake were reconstructed using environmental transfer model for iodine adapted to Belarusian conditions, and relative large data set on measurements results and activities in soil, grass, and milk samples were available. Also, thyroid doses for person who exposed to nutrient were estimated from results of individual thyroid dose estimated from mother, but also from calculation of this transfer coefficient from mother to fetus, in taking into account period of gestation and time of exposure. For estimation, they were taking different parameters. So sometimes the data were from taken from personal interview, consumption rates on fresh milk, milk products, origin of consumed foodstuff, interruption of consumption of locally produced foodstuff, residence at time of accidents, migration during April-May of 1986, which was acute phase of accident on the nuclear power plant, no? And also stable iodine prophylaxis, etc. So, and for this study, we took as observable variables hormone level of T3 and T4 levels in children at age, they were available only at age 11, 12. Also for other parameters, for example, IQ was measured in 3 points of time. When children had 6, 7 years old, is the same, like longitudinal study, no? 6, 7 years old, then 10, 11, or 10, 12 years old, and finally at 16, 15, 16 years old. Also, we had information about gestation week or quarter of the moment of the Chernobyl accident. And for this preliminary screening and exploratory analysis, we used MANOVA. We used MANOVA analysis, and also for descriptive, some non-parametrical correlation analysis for continuous data for both groups. Now not shifting.
Because it is right here. Try to do it from the
Yeah. No. What? Okay, you or it was you? No? Well, here you can see the descriptive statistics on thyroid, T3 and T4. You can see for control group, no, so they have higher no doses and exposed the level, and you actually have mean numbers here and median. Actually, for T3, mean and median was exactly the same, so no normal distribution here, slightly different. And now, no. Again, can you help me? Well, you just escaped again, yeah. And here for IQ, so we replicated that it was before also already verified by another study that actually for IQ at the age of 6, 7, 8, between exposed in utero and control groups, there was statistically significant difference, but not later. So in the ages of 11, 12, and 15, 16, this disappeared. And also you have here mean doses, no, for comparing doses. Only I can tell you that actually this difference is attributed to verbal part. Non-verbal part, there were no any statistical differences, but verbal part, yes. Can you help me to go to next slide, please? No? Okay. Well, and here some different model we are trying to test. You can go next slide, please. And here some results of MANOVA. And you can see that for group it was like in utero versus control. So if you see in yellow marked here, just paste it, no? So for IQ, if we are doing like MANOVA for the mixture, no? For the whole data set, we don't see any for IQ differences. But for T3 and T4, we have these differences statistically significant. As you remember from descriptive graphics, they were, you know, quite like half. And when we split it for dose group, we had 4 dose groups. On next slide, I will show you. But here you can see that for dose group, actually we haven't seen any statistically significant differences. Only maybe for T4, like marginal UCP value of 0.66. But for interaction, dose group by sex, by gestation quarter, yes, this statistically significant difference for T4 level differences was significant. Next slide, please. Yeah, and here you can see the doses, they were distributed. We had like 4 different group doses for microRNA per hour, 1, 2, 3, 4. Actually, they were split for more homogeneously for participants to be in each, you know, for number of participants for each group. But actually for more 20, it's like more considered a little bit high, you know, but the rest are very, very low, as you see. And actually for, when we compare it for this dose group for IQ, so the first dose group, like more than 20 microRNA, no, per hour, they had statistically different differences with the first 2 groups on IQ. You see, now BP value for 0.42 and 0.09. And the next slide, please. But for hormone levels, all groups were statistically different with all whole groups, as well as you see on the left for T3, as well for on the right for T4. So all groups were, so like each level of those group splitting was statistically significant for, it's very like sensible for hormone level. And next. I think we are finished. And here correlation analysis, but not for categorized data. It was for continuous data, since we are not normal, we're using not parametrical analysis. So it was like general also screening to see. And on the left, you can see control correlations. And we can see that IQ in controls were the statistically significantly associated with T3 level, with hormone T3. But for exposed in utero, this relationship disappearing. Moreover, it's changing the sign from positive to negative, you know. But actually, what we got here, that IQ is, well, didn't have any statistically significant relationship. It was marginal. I put in a little bit slight orange for T3 and for dose, when there is negative sign, but it's marginal. It hasn't reached 0.05. And only T3 here was shown in this group related to dose as statistically significant level, so like exploring data. Next slide, please. So for conclusions, we've seen that Inverse relationship was observed between T3 level and IQ scores in the exposed in utero group, but it hasn't reached, it was marginal level of PIT, so it hasn't reached statistically strict significance and control compared to control. And significant relationship was observed only in the control group with a positive sign. But thyroid hormone T3 had significant relationship with IQ in the control group, and doses were associated significantly in an utero group. So quite different final results. Next, that's all. I think that's all. Thank you very much.
No.
Yes.
So is it okay with the micro? Okay. Can I have? Yes. I'm sorry because if the title is a quick look of for relevant multi-generational and transgenerational effect studies, I can name it what you have seen.
Please
on-
go closer to the to the mic. Sorry.
Yes, excuse me, because I'm afraid by my voice. And yes, many things are.
Just close it because I don't hear online.
The mic up.
Yeah.
Yes, many things have been explained this morning. Is it okay? Okay, from the highlight of heritable effect of ionizing radiation, many questions have been asked, and it depends of the context of exposure. As tourism, we can be exposed continuously in for ecotoxicological study, it's interesting, or for population living in high exposed territory. And parents can be exposed before fertilization, before conception, or the last contact is in utero exposure. Then in the different context, for parental only germ cells, where somatic cells and only germ cells will be exposed, and for the last in utero exposure, the fetus and the germ cells of the fetus are exposed. So we expose both three generations. And after we can consider a transgenerational effect because no germ cells were directly exposed. So to resume briefly the state of art, for preconceptual irradiation and pregnancy irradiation, epidemiological observation relate no effect before fertilization, and after we can count many effects exclusively on brain system and body weight and some effects were related on the cardiovascular system. Oh, okay. It's me. About experimental observation, in female population, nothing was related before conception. And compared to male exposure, where carcinogenicity can increase and the lifespan of offspring decrease. We also think some neuronal and epigenetics effect were reported. Excuse me, this is a problem with my finger. During the pregnancy, from high to low dose, we can observe a decrease of body weight, brain, and a neuronal effect, behavioral and cognitive effect, developmental effect, report on liver, thymus, kidney, and spleen, many effects. But unfortunately, nothing is really clear at very low dose. And about chronic exposure, for high doses, we can observe some significant effect, but nothing below 20 milligrams per day of exposure at a low dose rate. So the conclusion is in utero exposure seems to be more sensitive than preconceptional exposure. This is why we are interested by this topic, I think. About the sensitive developmental period, as it was shown just before, by Kimberly. We have three major periods, the pre-implantation period with the all or nothing effect, and the organogenesis period, from which many effects can be observed. Posure. And when we can observe minor congenital anomaly compared to the organogenesis. About the all-or-one phenomenon, as the name precise, below a threshold, nothing can be observed, and behind the cell death by apoptosis, and there are no problem. But excuse me, a little problem. However, an experimental study observed some malformation at a low dose, and it asks the question: must we consider little risk during this first period? Because this is a huge social problem for women that they don't know they are pregnant. About epigenetic, what to think? So epigenetic is now taking in account in the risk of adverse effect, and because over context of exposure, stress, more than ionizing radiation, report an early stress during in utero period could occur to adverse late effect, a risk of adverse late effect. How does it work? You know this, what is epigenetic mechanism. But as he would say by Zdenko, epigenetics mechanism seems to be involved in the origin of development of many diseases like cancer and non-cancer disease. because an abnormal DNA methylation de novo in somatic germ cell may be a risk factor. So how does it work? We have a different sensitive period during the development, starting to the germ cells. Germ cells are programmed. There is a DNA programming of germ cells. It begins formal during in utero period at the fetal state, and more later for female. So this is why male can be more sensitive than female, depending of this age of programming. At the fertilization, DNA was cleaned up. This is a mechanism to clean up adverse effect, and it started just after implantation of blastocysts.
Can I ask you, so you said females develop later?
No, the DNA methylation programmation start after when in the follicle, very later from male. Male, the germ cells programmation begin in utero. Yes. And during this period of programmation, this is the most sensitive period for organogenesis that can impact somatic cells during the development of organs. And after, at the development of gonads, again, for the next generation, germ cells are demethylated and again methylated, like for the previous generations. So we can see this period of methylation can be considered as a very sensitive period. So, to resume, prenatal stress can imply an epigenome switch that can be transmitted to the phenotype and occur chronic disease later, and maybe that can be transmitted to the next generation. Alors, about transgenerational epigenetic inheritance, sorry. The cleanup processes normally try to clean up the past injuries. So how we can explain the possibility of inheritance effect? This is because DNA is not completely demethylated, and certainly some epigenetic marks carried by histone can be also transmitted through generation, and maybe the effect of small non-coding RNAs transmitted by spermatozoa that can impact the methylation process. In conclusion, I have some questions. If we cannot observe many effects at low dose, can we consider early stress as a potential factor of vulnerability? and risk for long-term health effect. And secondly, how can we consider the impact of endogenous and exogenous factors like pollutions, diet, hygiene, gender, genetic background, who are confounding or factor of vulnerability too, and which biological target and risk factor we can consider the general metabolisms, endocrine system, cardiovascular, neuronal, or reproductive system. This is the question on which system we must focus our study. Thank you.
So I hand off that microphone to Rafi,
Yes.
and he will continue with the radiation exposure for development and long-lasting effects. So that kind of fits. And I'll just say one thing that I've read, that if your mother was obese when she was pregnant with you, you have a higher risk of.
Please use the mic.
If your mother was obese when she was pregnant with you, any of you, you have a higher risk of colon cancer. So that's just an example of the milieu, the fetal milieu that we have. There are many, many things about that fetal environment.
Thank you.
So, to me, it's very plausible that many factors are involved, including radiation, in what happens to the fetus later in life, in adulthood, not just immediately at birth.
Yes. Okay, I have to go fast because I have many slides. So here I will explain mainly the radiation exposure during brain development and long-lasting persistent effects. So maybe I will skip the first slide about brain development, although it's interesting to know a bit these early stages of brain development. We will start from E7, which is just after the implantation period, what we call also early organogenesis, and we will also have interest into E11, E13. This is late organogenesis or the neurogenesis period. So I will skip this. So this is also interesting, but yeah, it will take some time to explain. This is all the steps of brain development, starting from...
Sorry, Rafi, your slide don't move.
Sorry?
At least I don't see. As your slide don't move, or is my status at least?
Maybe they don't see the slide changing. Sorry, I was in this slide. So as you can see here, let's say, sorry. The different processes are ongoing, let's say from progenitor expansion, neuron production to migration. So I will not explain much, but because we don't have much time. Yeah, sorry. It doesn't work again here. Once you use the pointer, yeah. Okay, thank you. So this is about the postnatal period, but I think I will not have time to reach this. We can go. Okay. Yes, thank you. So now we will go to the in utero radiation exposure. So we can see here the prenatal period, and in purple this period where, let's say, the cognitive effects have been observed from Hiroshima, Nagasaki, A-bomb data. So we will focus on these parts. We have exposed, let's say, embryos at different stages of development from E7 to E9 to different doses of radiation. Here I mainly presented 0.5 and 1 gray, and we examine at E18, just before birth, the malformations. So we can see different malformations like exoncephaly, open eyes, iris anomaly, agnatia, microphthalmos, anophthalmos. This is effect with the eyes, gastroschisis and cleft palates. You can see all with the malformations and the skeletal also staining that show the differences. Here the statistical values. You can see most of the malformation occurring irradiation is given at E7.5 with high rates of 62% of malformations. With 1 gray. When you see the difference with 0.5 gray, it gives only 13.24%. So these are really the highest values. So you can also see in these graphs, is the same data exposed differently, but just to show, let's say that E7.5 show mainly microphthalmos, anophthalmos as the highest malformations, and followed by malformation of the jaw and also the exoncephaly, and this is a malformation where the brain is out of the cranial box, and gastroschisis are all occurring at after irradiation at E7.5. In the other graph on the right, we can see that early resorption also occurred highest at E7, followed by E7.5, E8, E8.5. So there are also prenatal death. So looking to the molecular effects by using microarrays, we can see that most of the genes or the pathways affected are linked with brain morphology, eye phenotype, let's say optic placode or olfactory for brain morphology, also optic vesicle formation. Most of the pathways are related to brain formation or eye morphology. So, yeah. Is embryonic day. Yes. Sorry, I don't have to shout because so. So this is explaining the mechanism behind eye defects. So we could find that, let's say, ionizing radiation can block, let's say, a pathway that lead to eye depigmentation. So, but yeah, it will be difficult to explain it easily. In this experiment, we also provide folic acid before, let's say, pregnancy to the females, because this is also given to human, and we can see a clear improvement in the malformations, and you can see in the graphs of exoncephaly as well as anophthalmos and microphthalmos, all the malformation are reduced to a minimum compared to, let's say, the 1 gray irradiation when given 8 milligram per kilogram folic acid or 12 milligram per kilogram folic acid. So, but yeah, we can see that there is no difference between 8 and 12 milligram per kilogram folic acid. So also some, let's say, functional effects. We test, let's say, the optokinetic tracking response, and we can see in the control, let's say, without folic acid, that this is reduced depending on the dose. You can see 0, 0.1 and 0.5 gray. When we provide folic acid is only significant with 0.5, and the effect with 0.1 is removed. Also in terms of using MRI and measuring the eye diameter, we can see that with the folic acid, the reduction in eye diameter is prevented. So in conclusion, for this first part, E7.5 is the most reduced sensitivity day to elicit microphthalmos, anophthalmos, and exencephaly at 1 gray, while doses below 0.5 gray does not affect postnatal survival. Doses below 0.5 gray also at E7 caused adult sensory and brain defects, because we analyze also some behavioral paradigms at after birth. Folic acid was able to prevent many of the radiation-induced defects both at 1 gray and 0.5 gray. So now we will move to a later period in organogenesis. This is late organogenesis, E11, or at the onset of neurogenesis. So one of, let's say, something that have been discussed today, microcephaly is clearly demonstrated here. So with 1 gray, we can see a clear reduction of the brain size, and you can see in the graph below on the right the dose dependency, and this can start already at a dose of 0.33. At 300 milligray, we have already reduction in brain size. So we also analyze the behavior. You can see also a dose-dependent change in the behavior here. In the top one is the social behavior. So there we measure the capacity of the animal to recognize a stranger animal just by nose contact. So, and we change the animals, and so these are just to test the ability of the memory of the animal. And the test below is the Morris-Water maze, is quite known test to test for spatial memory, and also the strategy used by the animal to reach the platform. And there are quite some deviation. With MRI, we measure also different parameters of the brain structure. So in terms of the size, not only of the whole brain, but all of different regions of the brain. And we were able to make a correlation between the behavior and the MRI of the total brain volume and the latency, for example, to reach the platform in the Morris-Water maze. In terms of transcriptional response in these prenatally irradiated animals, we could find that P53 is the most responsible of many aspects as apoptosis, cell cycle, DNA repair. So, and also P53 induces premature neuronal differentiation in response to acute DNA damage during early neurogenesis. So, doesn't move again. Yeah, okay. Yeah. So here are some more detailed effects on DNA damage response. Yes, also cell division, proliferation, and migration. So all are affected, and here it's still with a high dose of 1 gray, but we also tested the 0.1 gray, and we can see a kind of dose response of these effects. Also, we tested, let's say, in P53 knockout animals. This is the top on the right, and we can see that the reduction in size is this time less with P53 knockout. That means is P53 the main responsible of the microcephaly observed. So E11, so one of the main mechanisms is premature differentiation due to disrupted adherence junction. And here, so we have, let's say, the symmetrical, asymmetrical division of stem cells. So yeah, I will need a whole afternoon to explain all the data. So in conclusion of this part about irradiation at the onset of neurogenesis, it induces cognitive impairment at adult age. Irradiation at the onset also of neurogenesis induces microcephaly. P53 knockouts show partial rescue of the microcephaly phenotype And this microcephaly is partly due to p53-dependent apoptosis and premature differentiation. More research, of course, is needed to identify countermeasures to prevent microcephaly and adult cognitive impairments after in utero exposure to radiation. And I will stop here.
Okay, so that's really interesting. I'm sure we could learn more. And we have one more talk, I believe, by Balas Matas. Right, sorry. I want to pass that and then just make one quick comment, which is that for decades, at least, our policy in the United States is to give folic acid to all pregnant women. I don't know how many other countries do this to prevent neural tube defects. All right, thanks.
Hungary was one of the first.
Was it?
Really?
I didn't know that.
Science.
Okay, thank you very much. So it will be a bit different. I hope not completely out of scope. But so actually, I submitted an abstract, and that is which come out. But more precisely, so why I think it can be interesting is that it looks so it want to show another aspect which relates the tissue architecture on the sensitivity to ionizing radiation, and which may be also relevant in case of in utero exposures. So, and first, we have here this assumption that we suppose a balance between cell death and cell division, which applies for multicellular organisms. And even from this point of view, we already see that there is a significant difference if there is a growth and development compared to the adult life, and we have a balance between cell death and cell division. And if we see the title, The Impact of Cell Death, if there is this balance.
Can't hear you.
Sorry, is it? Yeah, probably better.
Thank you.
It's terrible for me. So if we have an impact on cell death, that also means an impact on additional cell divisions. So ionizing radiation is a cell-killing agent, so it is a way of mutation induction, which may later lead to an increased cancer risk. So probably many of you know this figure from 2015. It was a rather famous paper arguing that most of the cancer incidence is due to just pure luck, because if we plot the total number of stem cell divisions in different organs and the lifetime cancer risk, then we see in logarithmic scale, which is quite important, then we see this linear relationship. But if we look a bit closer, then we see that they distinguish between lung cancer in smokers and lung cancer in non-smokers. So here we see that smoking indeed have quite an important effect on lung cancer. So it doesn't depend only on the stem cell division, but indeed there is some relationship. But what is important, and has been shown later, that there are very effective mechanisms which reduce the mutation load in different tissues. So if we imagine that we have a very effective branching of cell division, then it is not needed too many divisions to generate a rather large tissue. And of course, it's different in adult life, and we have to keep some self-sustaining stem cells, while in early development, it's more related to this kind of cell divisions. And that's the paper from here also arguing or debating the conclusions of the Tomasetti paper. And they also highlight that there is this Beto's paradox, which is an observation that at the species level, the incidence of cancer doesn't appear to correlate with the number of cells in the organism, which will be quite straightforward. So if the one cell division results in a given number of mutations, then you would expect that larger animals with longer lifespans will have a higher cancer incidence than others. But this is not the case. And this work would like to show that something similar can be observed in case of the tissues. So a larger tissue not necessarily have a larger cancer risk. Yep. So, and we also see some other examples of papers that we have some observations, biological observations of properties of the tissues, of the ratio of different symmetric and asymmetric stem cell divisions. And if we suppose that there is an adaptation against cancer or minimizing the mutation load, then we see that it can explain some observations. So what we can see in case of, and how all this is how ionizing radiation may affect all these processes, a couple of slides on that. So the classic view on mutation induction by ionizing radiation is that ionizing radiation increases the number of DNA damages. In this way, the number of misrepair DNA damages will also increase, so it lets an increased mutation rate. However, if ionizing radiation kills cells, and we have this balance between cell division and cell death, then the neighboring cells must divide more frequently to maintain tissue homeostasis, and this is another way of the increase of mutation induction. Yep. And what we did before, so in case of radon exposure, we set up a model, what is the contribution of these two ways of mutation induction to the total number of mutation induction, and we got the surprising result that the second effect, so the increased cell replacement due to the increased cell death rate, will result has a much higher contribution. This is the blue line compared to the direct DNA damages. What is important that here we supposed that for cell killing, the sensitivity of all cells was the same. So differentiated cells and more progenitor-like cells had the same cell killing probability in case of the same number of alpha particle hits, which is probably not the case, but it already highlights an important property of the tissue architecture, that even the sensitivity of differentiated cells to cell killing, to ionizing radiation, is an important factor which may affect the number of mutations which are produced in the tissue. So if you look here back, even if we kill some differentiated cells or more differentiated cells, then it induces cell replacement and an increased mutation load in the tissue. Yep, and another phenomenon, a couple of slides. So we see that there are mechanisms for minimizing mutation rate, and ionizing radiation induces mutations in these two ways. So if we look on cell death from a mutation induction or cancer risk point of view, then cell death may be advantageous because it decreases the number of cells with potential mutations. But because of the increased cell replacement, cell death may also contribute to mutation induction. So we may expect something in cell survival curves. And what we see, and maybe related to that, is this low dose hyperradio sensitivity and induced radioresistance. This is in survival curves, deviation from the linear quadratic curve, and it can be observed in many different cell lines. And we don't really know what are the mechanisms behind, but we see that there is an increased apoptosis at low doses, so in the steep phase, and there are intensive DNA repair at higher doses. And the most frequently applied mathematical model is uses parameters which cannot be validated independently. So if we try to set up a very simple model on how a tissue with mutagenic damages can eliminate these mutations, and we suppose that cells with the least number of mutations replace those with the highest number of mutations, then we get such curves depending on the parameter setting. So a simple model can reproduce the fundamental features of this phenomenon without introducing a rather arbitrary threshold dose rate. Importantly, there are two parameters of this model: the spontaneous mutation rate, which at least theoretically can be measured independently on these experiments, and the number of induced mutations per unit dose. Oh, that's a Hungarian slide. Yeah, and you can set up more sophisticated models, and which you can test with experimental data. So that is doing now, and so why it can be relevant for in utero exposures. Oh, sorry. So there are mechanisms which reduce cancer risk and mutation rate, even in adults, and most probably also in utero cases. They may well have a threshold dose, and this phenomenon can be an example or a manifestation of such mechanisms. Ionizing radiation results in a perturbation affecting and making it visible such mechanisms, minimizing the mutation rate. And what is very important, that the tissue hierarchy, the tissue architecture is very important on what are the effects of ionizing radiation. And yeah, it comes out also from this, but that's rather straightforward. Otherwise, that cells during growth and development results in a high risk due to the higher number of progeny and the lower number of cells which can replace the killed one. So thank you very much for your attention.
Can you get that last slide up? Yeah, can you bring up the people on the, like in a row since you're just fine? I guess we maybe have 15 minutes for questions, comments. Oh, we do? Sorry. I'm sorry. Klaus, please.
I was very impressed by the presentation of Rafi. Just greetings to Rafi. I haven't met you for a long time. He suggested that these changes in stem cell segregation might be a major factor involved. I didn't understand that. Could you explain it a bit better? Because that's certainly a very important point in your mechanistic theory.
Not segregation. Yeah.
Just
I didn't
whether you have
explain it.
Symmetrical versus asymmetrical.
Yes, yeah. But yeah, what we observe also is a defect in migration and what we call also premature differentiation. That means cells that are, let's say, start to migrate without getting the right maturation. So these are, let's say, defect cells that have been not eliminated from the system.
This I knew because I've known your work very well.
Yes, I know.
But just the stem cell factor, which was new to me, and I didn't quite get it.
Yeah. But these are new data, so we're working on that. Thank you, Klaus.
Thank you.
So I would like to comment your presentation, Stefan. Because you were talking about the mother, the stress in the mother. And I was reading a paper, it is from, I don't know, maybe 35 years ago. And they were exposing rats to the radiation, the moms, the dams. And then, so they have the control group that was not exposed, and the case group that was exposed to radiation. But because they were exposed, the mothers needed to be fixed with tape in a table and irradiated. And then so that they could know that this stress of being taped would interfere in the outcomes of the leader, they had another control group just to be not exposed but fixed. And they could notice that just this fixed thing caused the leader to have more extra ribs. So it was an outcome. So the stress of the mother caused the leader to have extra ribs. And I found that really, ribs, yeah. Just as, and I, stressed. No? Okay.
I'm sorry, that doesn't cause, just because you're strapped down?
No, but the stress of the mother, the stress.
Yes.
Oh, maybe in animals, but not in humans.
Yeah, in animals. Yeah. Yeah, I cannot find here because unfortunately I have security issues in my computer and I cannot. But
Variants of 11 and 13 ribs instead of 12, but
in rats, it was in rats. In rats.
I'm suspicious. Doubtful.
Sometimes the results observed in experiment can be curious, effectively, but not the contagion. If it's fixed. Rats dislike to be contaminated, and maybe a higher level of glucocorticoids that create huge stress to the fetus. No, I don't know. I don't know this article.
Corticosteroids and you know and bone anomalies, but I don't think so.
Yes. But we, in the study I show of E7.5, in the study I show with A7.5, we show some skeleton malformations, but yeah, in terms of ribs, if I remember well, because there are quite some data, there are sometimes reduction or fusion of some ribs. That's all what we observe.
From
From irradiation.
radiation.
Yeah.
Yes, pretty high dose.
Yeah, quite a high dose. Yes, exactly. Yeah.
Can I just be clear that Omid is not, there's,
Please use a microphone.
no, I'm just checking about.
Ah, yes, there is.
Or is that
Oh, I'm in.
pre-recorded?
Okay. You want to ask? Ahmed?
Thank
Ahmed?
thank you. I come back to the Rafi. Rafi, this statement about the activation of P53, as if we talked today for several times, is in the level of the gene because it's a late lasting, or is this because of the phosphorylation, acetylation of P53, which cannot be corrected, or at all in the epigenetic level? Did you check it? Did you test it?
Yeah, a good question. It's a good question. No, we didn't test it, let's say, for acetylation or other mechanism. But yeah, at least we, from a transcriptional level, we we know because when we use, let's say, the P53 knockout animals, so we have less effect on microcephaly. So that's the only proof. But at the level of acetylation, I agree with you, but it could be interesting to check.
Okay, thank you.